just want to thank the organizers for the invitation to give this talk today. Um, so I'm Timothy. I'm a PhD student in the Nabi Lab at UBC, and today I'll be presenting the story that we're putting together, where we're investigating caveolin-1 endocytosis in response to osmotic shock, and a lot of these the experiments that I'll show today is using STED. And I'll try to kind of highlight a bit through the presentation, kind of why we needed STED to really investigate and observe these caveolin-1 endocytosis. So a little bit of background first. So this story is really looking at endocytosis. So a little bit of background on endocytosis. Probably the most well-known best characterized pathway of endocytosis is clathrin and clathrin-coated pits. So clathrin is endocytosis occurs relatively quickly. It's been measured before that cell surface clathrin can completely turn over in around 1 to 2 minutes. Another pathway, which will be important for this talk, is the click and geek endocytosis pathway. It's been known to be responsible for a lot of fluid phase uptake in the cell. And for this presentation in particular, it's known to be necessary for the uptake of the cell surface protein CD44, which is a marker for the click endocytosis pathway, as well as the focal adhesion protein beta-1 integrin. Most importantly, and what I'll focus mostly on today is CAV1 and the caveolae endocytosis pathway. While caveolae really is one of the endocytic pathways, it's really different from the others. So it's most well known as with caveolae. Caveolae requires the protein CAV1, which I'll talk a lot about today. It also requires the adaptor protein CAVin1 to form these caveolae. So caveolae are actually very stable at the plasma membrane. So when you compare that to clathrin, while clathrin can completely turn over in 1 minute, caveolae at that same time would really only endocytose at a rate of around 2%. Furthermore, the protein itself, CAV1, has a really high half-life, so it's really stable, greater than 36 hours. So CAV1 itself is an integral membrane protein. So it can oligomerize, as I mentioned, into these caveolae pits that you can see by electron microscopy. That consists of, has been measured before to be around 144 CAV1 molecules. It can also oligomerize into a smaller non-caveolar S8S scaffold. This has been recently identified by cryo-EM. It's forming, 11 CAV1s are forming this disc-like structure. And we've done some work before, I just want to highlight, with single molecule localization microscopy, where we identified and labeled CAV1 in the cells. We were able to identify caveolae, as well as an S1 scaffold, which would correlate with the 8S. As well as, since we're labeling the cells, we're able to identify these intermediate size scaffolds, which we show are intermediate structures that are building off of this, the 8S scaffold, and would build into caveolae. So while that's what we kind of understand about CAV1 caveolae now, I want to take us back, give some more background about CAV1 research in the past that will kind of give you an understanding of the work that I'm presenting today. So back in 2001, CAV1 was proposed to internalize into a distinct organelle called caveosomes. So this was first identified as being CAV1 GFP positive, and in cells where you infect the cells with this SV40 virus. It was found that these structures were pH neutral, and it was also not at the time labeled for EA1 or lysotracker, so for a marker for the early endosome or lysosomes. A couple years after, in 2010, the same group ended up retracting that term of caveosomes being a distinct organelle. Instead, they found that what they suspect was that what they saw is an artifact of CAV1 overexpression as well as with the virus infection. Instead, what they determined was that CAV1 was undergoing ubiquitin-dependent degradation. So when they overexpressed CAV1, they were able to visualize CAV1 being positive for markers with RAB5, RAB7, as well as LAMP1 GFP. So these are early, late, and lysosomal markers. They also found that this process was ubiquitin dependent. So when they used this CAV1 mutant with the lysines mutated and unable to be ubiquitinated, they were able to demonstrate that this prevented CAV1 degradation. A little bit additional detail and looking more at cavin-1. So cavin-1 was that coprotein that's necessary for the formation of caveolae. Work has been done demonstrating that knockdown or knockout of cavin-1, also known as PTRF, resulted in the degradation of CAV1. So when you remove cavin-1, disrupt caveolae, you tend to degrade CAV1. It was also shown that when you, in this experiment where using knockout of cavin-1, CAV1 was actually being endocytosed, co-localizing with CD44, that marker for the click pathway. And this was suggesting that the disruption of caveolae to forming scaffolds would result in the CAV1 endocytosis via the click pathway. So more evidence is kind of coming out that there's a lot of crosstalk. It's not very crosstalk between these endocytic pathways, and it's not just a clear-cut individual endocytic pathways. Last bit of background is that more research has now been looking into other roles of CAV1. CAV1 being so stable in this, caveolae being so stable in the cell surface, people have been looking at other functions and roles of caveolae. And it was determined that caveolae could actually flatten, releasing scaffolds in response to hypotonic shock. And this was important for helping the cell to buffer buffer any mechanical stress and help the cell survive different mechanical stresses. So it's demonstrated that with hypotonic shock, or when you stretch the cells, you lose caveolae on the cell surface. Cavin-1 was demonstrated to come off, and then you were left with CAV1 scaffolds on the cell surface. So at this point, we were kind of interested in determining, kind of looking at caveolae, CAV1 under hypotonic shock, as well as determining what happens to these scaffolds without once they're flattened. So in this study, we're using, I'm using two different cancer cell lines. So the first one, the MDA-MB231 breast cancer cell line. So importantly for us, this cell line expresses both CAV1 and CAVin. So it has caveolae, which you can see in this. This is fixed cell 2D STED. You can see these puncta, which would likely correspond with caveolae. We're also using a PC3 cancer cell line, which expresses CAV1, but is genetically ablated for CAVin-1, so it cannot form caveolae. So here, the CAV1 labeling again in the PC3 cells, you can see it's a bit more diffuse, and these would all be, what you would see here is all scaffold structures of CAV1. So when we first kind of looked into this, did the study, we looked at longer term, long extended durations of hypotonic shock. And what we saw first by CAV1, by turf microscopy, a fixed cell with various time points, was that we were actually losing CAV1 from the CAV1 intensity. So we were suggesting that we were losing CAV1 from the cell surface. So CAV1 intensity was decreasing through time with hypotonic shock. But when we put the cells back into isotonic media, the CAV1 intensity would come back, suggesting that the CAV1 was returning to the cell surface. So then we did a 2D STED fixed cell imaging on the MDA231 cells. We were trying to determine what's happening to the CAV1. So with isotonic, kind of shown before, you get the puncta of caveolae on the cell membrane. But what we started observing was that at 15 minutes and kind of accumulating, quantified here at 60 minutes, we were seeing these spherical endocytic structures forming of CAV1, and they were quite large and could not correspond with caveolae. So when we saw these structures, we were then trying to understand better what they are, if we can see other markers. What we found out was that the click markers, CD44, as well as the click cargo beta-1 integrin, these structures were co-localizing with the CAV1 endocytic structures upon hypotonic shock. So with in isotonic regular conditions, CAV1 puncta, as well as cell surface labeling of CD44 and beta-1 integrin. But with the shock, we're seeing the beta-1 integrin and CD44 in these spherical structures. So by with knowing that, we then perform this antibody internalization assay to actually prove that these structures are being internalized and are endocytic. So what we did here was, because we were able to label, or we knew these structures contain CD44 and beta-1 integrin, and these are cell surface proteins, we could add antibodies to these two targets along with the hypotonic or isotonic media. And then when we did that, we would then, before fixing the cells, we would do an acid wash, which would strip away any cell surface labeling. And what you're left with is only the any antibodies that are internalized. Then we would fix the cell, label also for CAV1, and then label with our secondaries to do this imaging. So in this top left panel is a control. So the cells were, the antibodies were added in isotonic media for 60 minutes at 4 degrees. At 4 degrees, there would be no internalization or endocytosis, and you can see cell surface labeling of beta-1 integrin and CD44. When we do that acid wash, we will strip away any, basically all labeling of beta-1 integrin and CD44. If we do this at 37 degrees, you can see some labeling of CD44 beta-1 integrin, just reflecting some natural turnover or internalization of an endocytosis. But if we do this with the hypotonic media, 15 minutes and 60 minutes, and we see these, the red CAV1 spherical structures, we're able to see also beta-1 integrin and CD44 within these structures. So this validates that these structures are endocytic. This is quantified here, just showing quantifying the internalization of these of the two markers, as well as CAV1 overlap with CD44 and beta-1 integrin increasing with time. So next, we tried to further really understand what these structures are, what they're doing. And when we tested a bunch of markers, we then found that CAV1 was, these spherical endocytic structures were co-localizing with LAMP1 GFP, suggesting that these structures are late endosomes or lysosomes. So here, here is showing CAV1 under isotonic conditions, as well as LAMP1 labeling lysosomes. And then upon hypotonic shock, we're seeing co-localization of some of the structures of LAMP1 and CAV1. So now that we understand that these structures are LAMP1 positive, possibly lysosomes, we then went and tried to express the CAV1 ubiquitin mutant. So this was the mutant that was developed in that previous paper, where it cannot, this CAV1 cannot be ubiquitinated, and there's a decreased amount of degradation. But however, when we express this mutant compared to a wild type, we didn't really see a difference. This mutant was still able to form these spherical endocytic structures that were going into LAMP1 and lysosome structures. So ubiquitin was not necessary for trafficking to these structures. Then we tried to understand, well, what's happening to these lysosomes under hypotonic shock. This is a live cell confocal microscopy. So we expressed a CAV1 halo, as well as used the lysosensor probe. A lysosensor will fluoresce will be highly fluorescent in acidic conditions, and it's able to label lysosomes that you can see here under isotonic conditions. Upon hypotonic shock, we were seeing CAV1 forming those spherical structures, and this was also labeled for the lysosensor. So maybe at this point, I just want to highlight a little bit. This is confocal of these spherical structures. If you just kind of look at this structure, this image itself, it's not really as convincing as the STEDs, the structures that we can really visualize by STED. So we really needed the STED in a lot of those initial experiments really to be able to visualize these structures. We did notice that the lysosensor was much dimmer in these structures, suggesting that these structures are not acidic, that they could be pH neutral. So we also performed some controls where we treated the cells with baflomycin A. This prevents lysosome acidification. And when we did that, though, we were able to show that the lysosomes were no longer labeled like without the treatment. So we're preventing acidification, decreasing lysosensor labeling. But when we do the shock, these spherical structures still form and they're still dimmer. We still see dim structures by the lysosensor. We next really tried to validate what we saw here. So in this experiment, we labeled for lysosensor, LAMP1, as well as with CAV1. And we were able to show here that these three structures are labeling the same structures. And we see the decrease in intensity with the lysosensor. So this really validates that CAV1 is being internalized into these pH neutral lysosomes. So that was a quite a interesting result. We did look into the literature afterwards, and what we found was that with lysosomes, there was a 2020 PNAS paper that demonstrated that upon hypotonic shock, the lysosomes will actually swell up, and this is necessary for helping the cell to survive the mechanical stress. So it seems like our CAV1 is being internalized into these structures, and may have some function as well there. So next, we tried to really understand when the formation of these CAV1 lysosomes were occurring. So I previously showed you this graph. So we quantified that these structures were really appearing at 15 minutes post-hypotonic shock and really accumulating. We performed this experiment using a hollow monitor, which is doing live cell phase imaging. So with that, we were able to track cell volumes across time. So when we did that and performed the hypotonic shock, what we saw was in both our MDA231 cells, as well as those PC3 cells that don't have CAVin1, we saw that the cells initially, cell volume increased quickly upon hypotonic shock, but just as quickly and over time, the cells are acclimatizing to the hypotonic shock and really actually decreasing in cell volume and decreasing even further than their initial baseline cell volume. So this was suggesting that when we're seeing the formation of these CAV1 lysosomes, they're really forming as cell volume is decreasing and as and associated with decreasing cell volume. So with that result, we thought to test both hypotonic shock and as well as hypertonic shock. So in hypertonic shock, you would just get a decrease in cell volume. And so what we saw is with the 231s, as shown before, you form those spherical lysosomes. In PC3 cells, we can also we also see the formation of these spherical lysosomes. Really interestingly is that upon hypertonic shock, we were able to see in the PC3 cells the formation of these spherical lysosomes, but in the MDA231 cells, which have CAVin1 and have caveolae, we didn't see this occurring. We couldn't really, we didn't see any formation of these spherical lysosomes. This is quantified here where we did a count of those endocytic structures, showing with hypotonic shock in both the 231s and PC3 cells, we get the formation of these endosomes, but with hypertonic shock, only the PC3 PC3 cells were able to form these structures. So we sought to validate the role of CAVN1 and determine what it's doing, if it's necessary for the formation of these lysosomes. Here is showing this experiment in the PC3 cells again, that they can form in hypertonic and hypotonic media, but with the cells expressing CAVN1, we saw that in hypertonic shock, the cells were no longer, or CAV1 was no longer going into these lysosomes. Furthermore, we were seeing that with hypotonic shock, we can still get the formation of these CAV1 lysosomes. This is just kind of the similar image, just showing kind of two cells side by side. So the cells not expressing the CAV1 without caveolae, it's able to internalize CAV1 with hypertonic shock. But with the cells expressing CAVN1, which has caveolae, CAV1 is no longer being trafficked into the lysosomes. So here's just a concluding slide, kind of describing what we've found with this novel pathway. So we determined that non-caveolar endocytosis of CAV1 was occurring in response to osmotic shock. And there seems to be two steps to this. So in one, you need, if you have cells with caveolae, caveolae needs to be flattened and disassembled. So CAVN1 needs to come off of the cell membrane, leaving you with non-caveolar CAV1 scaffolds. And then in the second step, with CD44 and beta-1 integrin, there seems to be click endocytosis and CAV being trafficked, not really in response to cell stretching, but to actually be during the cells decreasing in cell volume, as well as or with hypertonic shock. And then what occurs is that CAV1 gets trafficked into these late endosome, lysosome structures that are pH neutral and LAMP1 positive. So with that, I just want to thank my supervisor, Robert Nabi, lab members, and I can take any questions.
Okay, thank you very much. We have plenty of time for questions. You can also drop them into the chat if you feel more comfortable.
Hey, Team C, nice talk. Just have a question about the reason why the shock treatment was chosen to see this phenotype, or did you see this similar phenotype with different treatment?
Yeah, so we were initially really interested in looking at caveolae kind of flattening, looking at scaffold structures, and really looking at that mechanism. And so we kind of almost stumbled upon these endocytic structures by chance. We do also, so when we saw the structures were forming with decreasing cell volume, with hypertonic shock, we do also have a collaboration with Phui Bun from Alberta. She provided us with these hydrogels, which could be, were created with different stiffnesses. And we were also actually seeing these structures forming with in low stiffness conditions on these gels. So we're still kind of working on that, really trying to validate that. But it seems that these structures are forming with kind of decreasing cell volume, decreasing stiffness in those kind of conditions.
Okay. All right, thank you.
I have a technical question. Just if you could comment on, your choice to use the Hollow monitor to look at cell volume versus other methods. What did you see the advantages of that? Was it simpler, lack of need for a label, things like that? If you could comment. Yeah, for sure. So we did try some like membrane labeling with like kind of confocal imaging, doing like a stack, Z stack. But yeah, it was just a lot more simpler doing it with a label-free just phase imaging. It was a lot more higher throughput. Like the software is kind of already designed to like segment a lot of cells and then output that data.
We did have a little bit of problems with the tracking software for that was not perfect. So we did have to kind of curate the data a little bit, make sure the cells were tracked throughout the time. But yeah, it was a lot more, much easier kind of doing it with that.
Satoru?
Thank you. This is great presentation. Every picture is amazing. I'm wondering, have you tested this? You showed the LAMP1 is coexisting with this caviarin. Then have you tested there any possibilities like this pathway through the orthicosome localization or directly transport to the lysosome? Because I wonder, I see seeing the picture, I see the size of the lysosome positive structure seems a little bit larger than my knowledge of
Yeah,
size.
for sure. That was like, I think the most challenging part of this project was trying to really identify what these structures are. So we tested a lot of markers, including Rab5, Rab7, early, late endosomes, but we didn't see anything there. So yeah, when we saw the lamp, it was quite interesting. As you mentioned, they're much larger. So we looked back into that literature, and that 2020 paper showed that the lysosomes were swelling with hypotonic shock. So the lysosomes, they demonstrated that there's a lot of like VRACs on and ion exchanges in the lysosomes, and they were swelling with water with hypotonic shock. And that was really they demonstrate there that it's necessary for the cell to survive the shock. They mentioned there that the cell membrane can't actually take so much stress. Like you can't have over 5 or 10% expansion of the cell membrane. So instead, the water seems to be going, the excess liquid seems to be going into the lysosomes.
Thank you.
We have a question in the chat. What materials did you use for your acid wash for the antibody internalization assay?
Yeah. So when we added the antibodies with the shock, then we for the acid stripping, I'm guessing that's the question. We used a potassium hydroxide solution that was that we acidified. And that's just kind of 2 quick 30-second washes, and that's able to strip away all that cell surface labeling.
So you mentioned a little bit in the presentation. I wonder if you could point out to us again, where STED was really, critical in this project. Maybe where you felt it was too much or you absolutely needed it, and if you had to adjust any of your STED imaging protocols based on working in these hypo or hypertonic conditions.
Yeah, for sure. Kind of to highlight that, I guess I want to show first like a confocal image. This is kind of live cell confocal, but you can see if you kind of, in the lyso sensor image, they're more complete. But if you just look at the CAV1, kind of looking at them enough now, I can kind of tell, oh, these might be like these spherical endocytic structures, but they're definitely not very obvious to point out by confocal. There's quite large, so you would think that like that they're easy to see. But some of them can be quite small as well. And this is also after deconvolution for both the STED images and the confocal images. So that really helps. Without the STED, we found that the signal to noise is really bad. So while you might see some of the very obvious, right, really like bright structures, you'll completely miss all these smaller, more dimly lit structures. So when we kind of started the project, it was really important because we didn't do 60 minutes of hypotonic shock at the beginning, and with 15 minutes, you don't get a lot of these structures forming. So I kind of had a hard time convincing even my PI that these structures were actually existed, because you see maybe like one of them in the structure, and only one of them are very obvious by without STED, without deconvolution.
again, if you have any questions, you can raise your hand or jump in, or drop a question in the chat.
Yeah, I'm having trouble finding the hand raised, but I'll just add to Tim's comment that at the beginning, it wasn't obvious that these were endocytic structures for sure. It wasn't even obvious that these were not just, oh, some organization on the cell surface. And by confocal, it just looked like the regular stuff we would see. And so the STED was really important. And then the acid wash, we're just talking to chat about this. And maybe, Tim, you can add just the reference to the acid wash to the chat so people can get it. And then we said, oh, these are really endocytic structures. So it wasn't really, it makes it, the STED really makes it a lot easier to define them and to be sure about what we're looking at. I think going back now, maybe we don't, now we do confocal, oh, we sort of know what we're looking at, but at the beginning it was really critical.
That's great. It was really like supporting the exploration then. That's awesome.
Absolutely, absolutely.
Great. Well, let's thank our speaker. That was excellent. And thank you very much for, showing how STED is so critical to supporting that exploration.
Looks like there might have been another question come into the chat.
Oh, okay.
Maybe not, maybe I missed it. Guang, do you want to ask that question?
No, I'm good, Nikki. Ask that question.
Oh, you did, sorry.
Yeah.
I missed that.
Yep.
Trying to keep up with the chat. Okay, did we want to do the breakout rooms?
Sure. So we have, these meetings, can run them until noon. I'm going to open up breakout rooms. We can, feel free to navigate and choose which breakout room we'll have. Again, you're interested in. Again, we'll have the discussion with the speaker. We'll have communications on microforum as well as just general discussion rooms. So I hope to see you in the breakout rooms. I'll open them now.
Sorry, I joined late. Is there, what is the discussions or communications on microforum?
Yeah, so at our last meeting, we were trying to figure out better way to continue the communication offline rather than in just these user group meetings. And one of the ideas that came up was working on Microforum to create a channel there. And I think if someone, we could talk about how to move forward on that front. For instance, if somebody wants to kind of, I don't know, head up that initiative, we can definitely talk about how to move forward on that so that we can keep these conversations going offline.
Thank you.
Hi, Ethan, how are you?
Hey, Carsten, nice to see you.
Yeah, nice to see you again too. So how do we actually, sorry, how do we join the breakout rooms?
Oh, so let me see. Is there a specific one you wanted to go to? Because I can, I think, move you into one.
Just move me into one, yeah.
Sure. Let me figure out how to do that. Can I put you in the general interest?
Sure. All right, thank you.
Thank you.
Hi, Ethan. I think I have a similar issue. I don't know how I cannot see the option to join a breakout room. I just
Okay.
you know,
Sorry. Yeah.
member hearing. I mean, and yeah.
Which one? So we have discussion with the speaker, the microform communications, or the general interest?
Maybe the general one. Yeah,
Okay.
thank you. Thank you, Ethan.
Thank you.